because you guys need to submit the report soon, right? So hopefully that you can actually have manage to run it, at least get some result to use it for your for the report itself. And also you have to familiar yourself with the steps, because next week Tuesday we're going to have a test, and the test, it will be a practical test. So you have to make sure that you familiar yourself with the run. So anyone managed to finish, or anyone having the problems? I remember yesterday Ian saying that you have some problem, right?
Doctor, still trying because of the server problems.
You still cannot log in?
Just now only can log in. I think I try and later I email you, doctor.
Oh, it's in your network issue or what?
I think it's my network issue because I come to UM then can use already.
Oh, okay. Because usually usually should be, because I can log in for here quite easily. Yeah. One thing I want to mention is that for the Go and KG, right, yesterday, let me show the screen again. All right. For yesterday, Go and KG analysis, right? So we use the Go profiler, G profiler to do the analysis, right? So in here it shows the result, right? And I would like, beside the plots coming from here. So two options you can do. One option is you select the top one. For example, this one, the top one is the protein domain specific binding, right, for MF. So then you check in here whether whether they are already listed as a top. Okay, now, so you see now here it mentioned the protein specific binding, right? So this one, it's one of the most enriched Go term. So what you need to do is you need to explain this in your result. You can say that top 3 Go MF term is these 3, right? And then you and what kind of what function they specify. For example, here is for maybe involved in photosynthesis, right? Involved in the chlorophyll, right? So you need to mention that in your result in these sections. And so, for example, here is show the Go term involved in temperature response. So this indicate that your differentially expressed gene, right, they are already involved in the temperature changes, right? And also photosynthesis is involved in the heat stress response, right? So this is what you would like to mention in your result. So one thing you can do, it's you can manually label the top 3. Let's say, for example, this one is top 1. So you can label it because this is actually the interactive one. So when you save it, it's quite tough to label. I'm not sure whether you can label or not here. No, you can only download the plots, but you cannot label. So I want you guys to label in here. So either using a line and to write down the term protein domain specific binding, and this one is for chlorophyll binding, and also this one is proton transmembrane transport activity. right? And here is organelle. So it lists the top three. So this one is response to temperature stimulant, photosynthesis, and also the light harvesting, right? And here are the protein. I mean the photosynthesis antenna protein and also the others, right? Those are involved. So some of the thing we can know is actually the pathway. So for example, now we know that this KGG map 00380, right? Tryptophan metabolism involved in here. So basically, you can go to the KGG map. Uh, 00, wait, 382. So from here, you can see that those are the pathway for tryptophan metabolism. It seems to be here it involved in tryptophan metabolism, right? I think in the FIPO folder, they were not able to highlight the genes involved. In other software, they will be able to tell you, which genes involved in which pathway here. And then they will be able to color it, to become green or red, indicating high up and down regulation. And then that will be easier for you to explain that result. Let me see whether I have some example. Not sure whether I have example to. Yeah, I don't think I have the example to show. Let me check online. Okay, something like this. Some of the tools, right, available. Let's say, for example, TV tools and others. They allow you to analyze the go in KGG, right, and allow you to import the data. So import your genes that involved in this pathway, and to here, to the this pathway, for example. Then, I mean, this one is actually manually, but the coloring here, the green and red coloring, right? Red indicate the up regulation, green indicate the down regulation. So for example, in here, you see that tryptophan metabolism, right, will trigger the auxin here, right? And then auxin will, lead to the up regulation of OX1, right? Subsequently to up regulation of TIR1 and others. Then eventually it will lead to the let me make it bigger. Okay. So eventually it will lead to the cell enlargement and plant growth, right? So this is a kind of pathway that I mentioned to this just now. You'll be able to highlight those boxes here, because each one of the boxes it's for the enzyme. For example, this one is a methyltransferase, right? So what IMMT, so the gene name, and they involve in not only the tryptophan metabolism but others metabolism pathway also. This metabolic pathway, this one is a general one. General, it means that it's a big pathway. You see, they are involved the entire metabolic pathway. I mean, all the metabolic pathway of an organism. So you can see they are so complex. There are more than one pathway. So you have the Krebs circles and all others here, right? So this is the entire pathway. And then you can break it down to smaller one, tryptophan and others, right? So those are more focused, more defined. So for example, this one only this pathway, and you see that this genes it's involved maybe in here, right? So by using the I think one of the R program, you can actually plot this kind of patterns, and then you can manually put in your other information here. So you can either put in like this, or you can put it like you can even draw it yourself. Let me check. The reports. So for example, in this one, in my student's paper, you can actually draw this thing. So draw the pathway, you make it simplified, right? From just focus on this one, right? And then from tryptophan metabolism, right? It lead to the auxins, and then upregulated the auxin 1, right? OX1 and then TRS1 and others eventually lead to the cell growth. So what are those color box here? Those represent the matrix heat map that you saw yesterday. So the one that you saw in heat map. Yeah, this one. So you can actually find out the genes, right? And then extract the information and put it into something like this. So the first one, it's actually the control, and this is drought treated. So you can compare between 7 days and 14 days, and you see the expression changes, right? The white color one, it means that no expressions. At the 14 days, then the expression change of the OX1, right? One is become upregulated, one is down regulated. So because it have more than one copy of the gene isoform. So this is how you can actually explain. In order to get the self enlargement and plant growth, those genes listed in the pathway, right? They need to be upregulated, right? In order to have that. Same thing for here. So this is a kind of the pathway that we are talking about. Every time when we talk about Go and the KGG map analysis, right? So Go usually is only for the terms, when you have let's say for example, maybe here, for example here. So this means that you have several genes, right? 300 several genes involved in the response to temperature stimulant, right? So this one you have 1,010 genes involved, and this is 367 genes involved in that the response. So those genes, they usually share the similar expression profile, right? And here indicate that 20 genes was involved in the photosynthesis of antenna protein. So you can actually open up this map. Yeah, so this is the map in the KG map, and you see the genes involved here, and also the light harvesting chlorophyll protein complex. So you will see the different expressions. You know, there are transmembrane activity here, right? And how it regulates the photosynthesis. So from here, then you can know uh, about the expression profile. I just need you guys to mention the top three. You know, what potentially involved. So for example, you can look for let's say in other plants, heat stress. This is, mm. So some of the information from this journal, right, thylakoid proteins, and they involved in this. So you can refer this one to find explanation to support your finding. Now we know that the genes in the arabocyst under heat stress, right, they are regulating this or involved in this photosynthesis antenna protein pathway, and you need to find the temperature. I mean, you need to find the support, right? So you can look for the articles. So basically, the keyword usually you want to check is, you can put either something like that is, the stress that you are studying, right? The pathway that involved. Then when you search, right, then you'll be able to know. I mean, this is by AI. They're showing that involved. Then you need to check in whether the extraction, I mean, the AI extract the um, information from this paper or not. Is it clear or not? Right. If this is correct, then you can use it to support your discussion. Right. And they provide some explanations why heat stress, cause minor antenna protein to detach from P2, and then what happens, right? So in here, yeah, you can read about, for example, this paper here, right? And the consequence of the on the photosynthesis will involve energy imbalance, right? And all others. Of course, check the paper, right? How photosynthesis respond to high temperature stress. So this is the kind of the explanation you will provide in your discussion. You know, you mentioned that there are these pathways involved, and how this pathway affect how regulate the high response of the plants against the high stress. So how they regulate here is showing that provide explanation how we regulate that. Yeah. So this is a kind of thing that we will usually explain in our discussions, and also the result. Result is just actually result is just mentioned the three main term, right? Photosynthesis, antenna protein, and protein processing in endoplasmic reticulum, right? And also the photosynthesis. So it means that those genes involved were enriched in these three different pathway, right? And then how they regulate or interact within this pathway, then you provide the explanation, something like that I explained here on top. This is for the transcriptome. Yeah. There's another way of running that go in KGG is to use the ENOC mapper. So this one, it's actually by blast. You would put your protein of interest. Let's say, for example, now I put the Arabidopsis protein, right? Or the protein, right? Then I through the blast, right? Blast P in this ENOC, ECNOC database, then I will be able to get the result. No, this one here. So then I will be able to annotate each of the genes. For example, this gene number, this gene, right? They involve in, it is a NADP dependent enzyme, right? And then they involve in different, it is the enzyme ID, right? And what pathway involved. So this provide the information, and also what is the protein family they belong to. So ADH family. So this is for you to annotate the your differentially expressed genes. Right, assume that this is a differentially expressed genes. So what you need to provide is actually you need to provide the peptide sequence. So you provide the peptide sequence to, right, and then go through the run. So you use a sensitive mode. Yeah. And that allow you to, and also the minimum E value is 0.03. This is a E value usually for blast, right? And you need to set the taxonomy. This one is for VD planting. So what is the taxonomy ID of this one? Yeah, it's for VD planting. This one is for plants. Since we know that it is Arabidopsis thaliana, so it's belong to green plants, right? So we need to input this for the ECNOC mapper to run. So they will only blast against the genes in all the plants rather than others, right? Then output option, you will have output all, right? This one is not necessary. Then you produce the 3 different file, and this one is the autologs. For example, for these genes, right? So what is the autolog name? Both are autolog. So in Arabidopsis thaliana, basically more important is this one, because this one later then you can use the TB2, you know, to draw the analysis, to draw the to draw this kind of plot in TB2s. Yeah, but I'm not going to show this one because by doing analysis using the G folder, I think it should be sufficient for your report. Because as long as you show this one with the manual labeling of top 3, then I think it's sufficient, and explanations of those involved. Yeah, just need a simple sentence how they involve in here. So let's see the discussion of signal transductions. Let me find it. Yeah, so provide insight into how a pandan adjusts its functions and metabolic pathway when exposed to drought stress. So then you provide explanation on the, under the heat stress, goal analysis show up relation of DG in this activity, right? Indicate that enhanced gene regulation and nutrient transport. right? In terms of plant physiology, transcriptions, factors, something, this, right, provide explanations for the Go and KGG. And also you mentioned about plant hormone and signal translation and MAPK pathway, right? So they consider and enrich between the drought stress, indicate a continuous signal adjustment in managed to stress response. So, and this also has been reported in other drought stress plant, right? And also just now you see that auxin signaling pathway play an important role in helping plant adapt to the changing environment, right? As shown in the auxin, yeah, this one, right? Auxin response pathway. So you provide an explanation for that, and you see that many of the genes fall into these three major family, right? And the OX1 expression, right, exhibit a complex region, right? One transcript upregulated and four downregulated at day 7. So it's this one here. Yeah. So you can see that one upregulated and four downregulated in day 7, right? When you move to the day 14, the expression profile will change, right? You see many were downregulated. So this kind of explanation you need to provide actually in your discussion. Just simply one or two sentences, it should be sufficient, right, to support your finding. Yeah. Okay. Any questions from you guys? Any questions?
Yes, can you please repeat on how we do the protein on Galaxy? Like how do we run it?
How to? I didn't catch you because it was not clear. Sorry.
How to run the protein on Galaxy? Like how do we get the file again? Can you repeat that?
Which file? Which file?
The the one that show the top.
Oh, you mean how to get this one?
Yes.
I mean how to get the KG map or something.
Yes.
Okay, for example, now we know that in here is KGG00196, right? So this one is involved in the photosynthesis on antenna protein. So in the Google, you just type the map 00196, right? Then you will get able to get the map information. So the map here. But anyway, for your side, you don't need to have this one. Because why? Because even you're able to get the map, right? You don't know which gene is involved. The reason why, because the Go profiler itself, right? They didn't tell us which gene is involved in here. It didn't list down. It just do a summary on it. So that's why we can't know. I'm not sure whether they will allow us to check. Let me see. Let me see whether would he be able to provide the genes. I can't see anything allow us to get the gene name. So because it's only they put it as a summary. So I can't know. Let's see whether is there any other tools that I can use. No, to show which one is actually OKGG. Oh, this one no. Mm. Mm. Cluster group gene category connect by share genes. So this is a FASTA file. Mm. Mm. Seems to be not able to cluster them in here. No, I can't find it. I thought it can show how to plot the image in Galaxy, to plot this type of image. Yeah. Huh? To plot this type of image, or yeah, something like that. To have this kind of image in there, but since they are the G profiler not allow you to do. So basically, you just need to mention that which pathway involved, and then what function they involve in here. So in here, you see that KGG, right? KGG, I think it's at the lower part. Okay, for example, this one. You want to know how the genes involved in heat stress, right? Yeah. So for example, heat stress disrupt the ER homeostasis, causing protein to misfold, right? So that's why you see this the genes is involved in this protein processing in the endoplasmic reticulum, because heat stress it destroy the protein, right? So then it activate this. So this is the explanation why you see the pathway of this term was identified in your Go engaging analysis, because heat stress, it changed the protein folding, right? And then, for example, how heat stress affect the photosynthesis pathway. Yeah, so because heat stress disrupt, for instance, that damage the thylakoid membrane, then you can check whether in here you're able to find the thylakoid membrane. Yeah, you see. So it affect here, and it also affect the cell cell component of photosynthesis 1, as mentioned here. So this is here clearly show that heat stress, right, that affect the thylakoid membrane, affect the photosynthesis 1 pathway, and also affect the chlorophyll pigment, because in the result here, it also showing that they involved in the MF molecular functions. Yeah, protein domain specific binding, chlorophyll binding, proton transmembrane tractable activity, right? And this is all most of them actually involved in photosynthesis. So the main thing is the heat stress actually disrupt the photosynthesis event, right? So from here, then you can show that finding in other report, other plant species also report that. And this is the kind of explanation you want to provide for your report itself, right? To find the enriched term, for Go and KGG, it's not difficult. You can use a profounder, but to find the explanation or supporting evidence to support your claim, right? It's sometimes it may not directly show in your result. So you need to try to find the paper to support that. Yeah. But for example, in this case, it's really show that it's how the genes involved in response to temperature stimulant, right? It's the one that's differentially expressed in your data, because it is high temperature stress, right? And now we know that photosynthesis also involved, because high heat stress disrupt the photosynthesis, right? And disrupt the light harvesting photosynthesis. And once the photosynthesis disrupt, it will maybe affect the ATP biosynthesis process also, right? And also when it's involve light, it will involve proton, right? Yeah, and also involve the transmembrane transport of protons. So this is all correlated to each other. So they are all under the photosynthesis pathway, as you show in here, the KGG pathway. Yeah, photosynthesis. Of course, there are other pathway. For example, the metabolite pathway. This one is auto photosynthesis, I think. This another one. This is secondary metabolite pathway. Yeah, those are the secondary metabolite pathway. So they're also involved in here, but I only want you guys to list down the top 3 and provide explanation why you think that's how gene involved in this pathway respond to heat stress. So the explanation is something like I mentioned here, how they respond to the heat stress, or how heat stress induce the genes in this pathway to become differentially expressed. Yeah, something like that for your discussion. Okay. Any others questions?
Doctor, so for the transcriptome report, we will need to find the differentially expressed genes and then explain why in that particular situation the gene are expressed that way, right?
Yeah, you see, now you're using a DE6 pathway, right? Then you know the differentially expressed gene, right? Like yesterday I showed those the differentially expressed genes. And you take the differentially expressed gene ID, you put it in a chip or folder, then you will know the pathway of those genes, right? So why you want to find only the differentially expressed? Because you see, those are the one that induced by the heat stress. One possibility is because those genes is induced by heat stress. It did them to become differentially expressed, right? Or those gene is differentially expressed because the Arabidopsis respond to heat stress. So they differentially express the genes, make them responsive to the heat stress, right? For them to survive under the heat stress condition. Yeah. So there's a forward and reverse direction, right? So forward is something like, heat stress induce the differential expression of those genes, right? And differentially express of those genes, it leads the plant to be survive better under the heat stress conditions. So this kind of regulation. And in order to understand whether the genes up or down regulated, right? How the upregulation and downregulation of genes lead the plant to respond to heat stress. You need to understand what those genes belong to. Belong to what pathway, belong to what goal term. So it means that which function they are involved, right? So that's why you need to perform goal and KHG. And then once you got the result on goal and KHG, right, you see they are under different pathway. Then I know now, okay, now these differentially expressed genes, they are involved in photosynthesis pathway, right? So now I know, okay, now they are involved in photosynthesis pathway, but how photosynthesis pathway respond to heat stress? Then you need to find the support, right, from other people. So as I mentioned just now, it showed that, right, heat stress itself disrupts the photosynthesis by disrupting the thylakoid membrane, right, and then affecting the photosynthesis one. So this affect the photosynthesis process of the plant, right? So what happens if photosynthesis of plant affected? Plant will not have enough energy, right, and they may not survive. They'll die from there. So this kind of explanation, it's showing that how the genes in a particular pathway, regulate or respond to the stress. Okay. You get what I mean?
Yeah, doctor.
All right. So this kind of explanation, basically you are trying to tell the story. Okay, now I saw this differential expression, right? So why they are upregulated, why they are downregulated, right? So for example, the one that I just show in the panel here. Okay, I see all the gene is upregulated. Let's say, for example, the gene is upregulated, right? For auxin 1 upregulated, it lead to the upregulation of TIR1, right? Then it lead to the inhibition of auxin X, this as we show in here, right? And both of them is a transcription factor. So they regulate the transcription of the DNA element, right? Then that lead to the changes in the expression of downstream gene GS3 and SIR1. So what happened then? Cell elongation and plant growth will be affected phenotypically. And in here, stomata closure, right, seed dominancy will be affected, right? Under the high drought condition, stomata will close, right, to protect the plant for water loss, right? If this happened in the seed, what happened then? Seed have not enough water, right? So they will not be able to germinate. So this is a kind of thing that you will provide explanation from when you're understanding the pathway here. Right. Yeah. So, and those are the differentially expressed gene as you can see here. So this is how differentiated expression, in a particular pathway. It's easy when the differentiated expressed gene is in a pathway to explain the, cause and effect, right? When you have a genes, you see a clear differentiated expression, let's say 30-fold up regulation, right? But the genes do not involve in any pathway, then that will be difficult. You need to design your experiment, differently. What you need to do is transcriptome itself might not able to help you. Now, you will need to knock down the genes, overexpress the genes in arabosis to see which pathway was affected by the knockdown or overexpression of the genes. Then from there, try to, make a story. Let's say, for example, overexpression of these genes, right, it lead to the downregulation of the carotenoid biogenesis, right? Then you know that, okay, this gene is a silencer because overexpression of this, the gene A, right, it lead to the downregulation of this, let's say this gene, right? Then it involved in the carotenoid biogenesis pathway, right? Then it affect the downstream process. So it affects the stromata closure. So what will happen then? The plant will become sensitive to drop. If the upregulation of, let's say, gene A, right, it lead to upregulation of these genes here, right, then it may be they are the transcription factor that promote the activation of PYR and this PYL genes, right? And then it lead to the stomata closure and then provide drought tolerant to the plant. Yeah. So those are for the genes that is not in your it's not listed in any pathway. They are more difficult to study because you don't know whether which pathway they belong to, right? You don't know whether they are enzyme or they are transcription factor or some others. So then you have to think of how you design your experiment, to understand that. When you have a genes that clearly show in the pathway, right? Something like the, in this kind of pathway, then it's easy for you to explain it because you can explain easily. Like, for example, downregulation this, right? Yeah, you lead to upregulation of this and others. And then what will happen then? You will lead to maybe others activity downstream, right? Then it will be easier for you to explain it. Let's see whether I have better example. This one cell cycle. MAPK. Too complicated. So for example, let's see we see about this one, right? Under the ERK pathway. So let's say under the stress condition, it lead to the downregulation of this MBK5, right? Downregulation of these genes because it's involved in the photosynthesis, phosphorylation of the protein, right? It also lead to the downregulation of the ERK5 and eventually lead to the downregulation of this NUR77, right? What will happen then? It affect the transcription activity, right? It lead to the proliferation disruptions. Okay. See whether I can make it. Wait, it affect the proliferations differentiations of the activity, right? So this is how by understanding the pathway, right? You know the side effect. You know the effect of the pathway, right? And then you can design your wet lab experiment to see, do you see any proliferation, differentiation being disrupted because of downregulation of these genes? Yeah. So some it won't lead to the apoptosis, right? So how the downregulation of these genes, right? So here you see that the MAPK signaling pathway is significantly enriched for DE genes, right? So means that DE genes are involved in regulating the MAPK signaling pathway, right? So you see that genes with the significant differentiation in tissue are shown in red. So those are the generally involved in this Roman tissue, right? The others is the one that in general expressions. So you see many gene is downregulated here, because green color usually mean downregulated. Yellow it means you have two set of gene, one is up and one down, right? And then red is means upregulation. Yeah. Some others example. Okay, maybe this one. So you can see here now orange is down regulation and this red is up regulation, and then yellow is the you have up and down regulation, right? So from here you can provide, okay, see here the up regulations, you know, the the stress treatment itself involved in up regulation of this gene, right, lead to the up regulation of this gene, but somehow it lead to up and down regulation of these genes, this one. So it means that there must be another pathway, you know, lead to the down regulation, because by looking at this, right, the stress itself indirectly upregulate the genes, as shown by the dash line here, and then the upregulation of this gene MEK5, it lead to the ERK upregulation, right? Eventually, it's supposed to be upregulated and NUR77, but it's showed the yellow. It means that there's another pathway that lead to the downregulation of the same gene. So that's why you got this mix, right? So and it here also indicate this one is actually a gene family. So you have more than one copy of the genes, right? And then it leads to the transcription activity affected, right? Because those are the transcription factor, right? So they affect the transcription, right? The affecting the transcription of downstream gene that's involved in proliferation differentiation. So that's how you'll be able to see that. Yeah. So you can see that in this line here, those are all the transcription factor genes that's regulate on the DNA, right, and lead to the transcription activation or silencing. Yep. Okay. Any more questions? Or you have any question regarding your run? You have any problem with the run or any type of run that you are facing? For for the repeat masker, I think as mentioned before, yes, I mentioned yesterday, you can change the version, right? So sometimes I think it's because of the latest upgrade of the Galaxy version, right? It lead to the problem. So once you change it, then you should be able to get the result, right? Okay. Any questions? Try to run the analysis. Try to by yourself. Complete the analysis. Yeah. Even though that let's say, for example, you are in a group, right? You only submit one report, but I hope that all of you can run, test run the pipeline so that you know how to, analyze. If I give you one data, and then you can help to analyze the data and then to explain to me, or even to your supervisor, because I'm not sure whether you guys what kind of project you choose for your your FYP or something, but a better understanding in the bioinformatics I think is important. For me, I personally think it's important, especially now because we are dealing with the big data, and to make sense from the big data, you need to have a strategic planning, so that you can explain the data, especially in the transcriptome part, which gene is involved in what pathway, and how this pathway affect the stress, or how these plants respond to the stress by regulating the gene that involved in a particular pathway. Yeah, this kind of thing that is, and then you know that you find a target gene, right, for your study. Then you try to manipulate the genes, by changing, by silencing them, by overexpress them, by knock down and knock off and others activity, right, to understand the gene function, to understand how this gene regulate and host in response to the stress. All right. Yeah. You can show. Can you show your screen?
Yes, Doctor. In a moment.
Yeah, let me stop sharing first.
Can you see them, Doctor?
All right. So what is actually the issue?
I don't really know because I've tried to run this. I mean, like
Can
after
you-
yes.
Can you refresh use the refresh button of your Genoscope? No. Yeah. I want to see your input. How do you run your Genoscope? The refresh button there.
Here,
Yes.
right?
No. In the cursor, the rerun button. Not this one. Up there. You see there's a circle. The arrows next to the box. You see the box? There's a box there. Right?
The box. Where is it?
There's an insect box. Box. Down. Go down.
Here. Oh, this.
Yeah. Run the
Oh.
job again button.
Right.
Yes.
Sorry.
So what is that issue? Your run 21. I think it should be correct. And can you click on the bug?
Here, this one.
The one. No.
This.
Yeah. I want to show. I can't see. Can you move a bit down? What is the error?
Here, is it here?
Error in significant number.
Or is it because of my previous run? Is it?
You
Right.
put your input. You putting the
The histogram one.
histogram.
This file.
Yeah. It should be correct because your histogram. Can you put the I button on the histogram? Press on the about. Yeah. Yeah, it looks okay. Your histogram looks okay. Maybe it's the you need to change the type of the file. You go to the next the pen.
Yes.
Yeah. And edit and see. I want to see what is this format.
It should be tabular, right? Because before this in a TXT format, and then it did it. So I changed it to the tabular, but still like this.
The original file is the TXT, right?
Yeah, originally it's the TXT and then I change it to
So
the
did you
tabular.
run using the TXT?
Yeah, it
It's-
cannot read at all.
Okay. Okay. So then in here, tabular is still giving you the error.
Mm. For the TXT, it cannot run. For the tabular, it's error.
Okay. Can you go to the run again button? Press again, run again. Yeah, this one I want to see.
For the jellyfish or for this?
Genoscope. Yeah, this one, yes. So histogram is correct. No, I want to don't press then run. I want to see your setting. Just
Oh,
want to see the setting for press run the run again button. So then I know your input setting. Yeah, okay. Wait, already go down a bit. Output. That's correct actually. Nothing wrong with your. Oh, then can you try using other version of Genoscope?
other version of it.
Yeah. Next one.
This.
No. In the middle, this one. You click here. Yeah, you use other versions. Let's say this one.
Oops. This one too. So and run.
Yes, and run and see.
Okay.
Because sometimes maybe it's a version issue.
Mm.
Yeah, you try and see whether whether you can get the result sooner.
Okay.
Maybe it's a version issue or sometimes because especially no then it's the histogram, then it should be your histogram or others because
So it's because of my previous.
the previous run, those.
So I have to run it again from the interlacer, I think, or is it not?
Yeah, I think your interlacer is correct, right? Can I check your interlacer and use the run again button?
Is it here?
Ah,
Wrong already.
yeah, because you see
Okay.
your R2 pair and R2 pair. What is this R2 pair and R2 pair? Right.
Uh-huh. It should be.
I
It
think
should be
your trigonometric also wrong already.
Mhm.
You go back to your trigonometric and press the run again button.
Okay, where is that trigonometric?
At the bottom. Yeah. Okay.
Which one?
Either one.
Okay.
Yeah. So you press the run again button. Yeah, you see.
Wrong already.
You see your input FASTQ file. R1 you put 1, right? R2 you put 1. Both are 1. Wrong, man.
Because I followed the manual.
No. It's
Oh,
R1 and R2. So it's supposed to be 1 and 2.
okay.
So that's why it's wrong.
Okay.
So actually
Because I was confused too because it should be different, but then
it should be different. It should be different.
It's the same file. They put
Yeah,
it
but in the manual, which manual?
the first one. Where is it?
Is it? Let me check. And
It's in trigonometric. Trigonometric is in the first one, right? But it's the same. It shows the same.
wait,
Same file.
manual. In the trigonometric part.
Yes.
Let me check. Trigonometric. So fast QC. Short read.
So it's in here. I have to change this one.
No,
No,
wait, where is the fast trigonometric part? Where did I put the traumatic part? Wait,
Or is
Which-
it that I just read them wrong? Because sometimes I think I'm right, but I'm wrong.
wait, I need to find the manual first. Where is
Okay.
I myself cannot find the manual. Assembly filter. Oh, I didn't put the traumatic in the manual. Is it? Macam tak ada.
Really, then what did I follow? Okay, but
Macam in my practical manual 11 tak ada pun, and in my practical 10 also I didn't put lah.
oh really? Okay.
Am I? Let me check again. Macam tak ada letak. Looks like no. Yeah, well. Oh, got.
It's in
Wait,
the second manual.
Yeah. First manual actually. Let
Oh,
me
first
zoom in first. I can't see. Yeah, because it's too small. Oh, yeah. Here it's the example only. So yeah, because you need to understand one thing is because R1 it means the one, right, correspond to the file number 1 forward. R2 is reverse, so it should be different. Yeah.
Okay.
I think this one it's only the mistake because of that. Yeah.
Oh, so it carry forward the mistakes and all.
Yeah. Because
Okay.
that's why I explain. I usually don't ask you guys to follow the manual because I explain it, and you just follow. You know what I want is like when I explain in here, right? You guys can follow. You know the steps rather than referring back to manual. It's just for reference only. Yeah.
Okay, doctor. So I run it again and I'll try again.
Yeah. So your problem is because you use the wrong setting. Yeah. Also my problem because I didn't check that properly. Later I'll try to change it.
Okay, doctor. Thank you, doctor.
All right. How about others? Okay. So sometimes when you have error, right, in one of the run, maybe it was like in this case, it was because of the previous step wrong, then it lead to the subsequently you can't analyze. So this is how you see how sensitive a pathway is. Tool is this. If you do something wrong in the upstream, you might lead to downstream problems. Yeah. All right. Any others questions? Oh yeah, one thing. Let me show again. For the Go portfolio, right? The first time that I ran, I did not provide the background, right? So I think it's important to give the background. So what is the background then? Background file. The background is the gene that is expressed in a particular data set. So this is here from normalized here. You need to take the gene ID of all the run, all the gene here, right? And you make it as a list like this. Okay. You make it like this, right? And this is your background. And then the DE file, right? DE file. In this DE text, it is actually among coming from among here. You need to identify the one that is differentially expressed. So how can we know which one is differentially expressed? Right. You will need to take the result from here. Right. Identify the differentially expressed, filter out those that is differentially expressed. Like I did yesterday, right? I download the file. The one that is from the differentially expressed result file here, right? Then I copy this one ID. So insert up. Right. Then I want, oh, this one is J. Okay. I want to remove those that non-differentially expressed, non-significant first. So I remove those that's, bigger than 0.05. These are the one that is, you need to delete them, right? And then I do the sorting. I sort the data sorting and using the log FC, smaller to largest. Then I filter out, remove those that is bigger than -1. So this one is bigger than the -1. I don't want this, and smaller than 1. So those are the one that is, they are differentially expressed, but the threshold is not the one I want. So I have to delete them. Right. You delete from here. Then this is what we call the differentially expressed gene. There are 100 something of them, I think 165, 64, right? This, so you copy the gene ID, put it into the notch PAD. And then replace the genes, remove the gene actually. So then save this as de.text. Then, right, upload into Galaxy. Yeah. For the upload it into Galaxy. Right. And for the background, you need to download this file, right? And then get the ID or ID from here, from column number 1, and then remove this and use this as a background. Yeah, for your GoPro, this is a background. Use it for your GoPro folder. Yeah, in here. Right, then you can run the analysis. So you can see the one that involved in organelle. Organelle is chloroplast is one of them. Photosynthesis 1 and thylakoid, those are photosynthesis, right? So response temperature, and this is the one that we want, right? Compared to the one that's without without giving the background, you see there are more involved. So top 3 is still the same, but others there are more. So those are the not the correct one, because this is involve everything. Right. Okay. All right. No, it's not MCQ. It's online. You can do it online. I'll give a data set. Each of you having a different data. Then you need to extract the data set. Right. each of you will have different data sets. And then I will ask the questions. Then you need to answer the question based on your result. Let's say, for example, I ask which pathway was affected by the, in this case, for example, heat stress, right? Then you need to show me your outputs, like this case. So you can explain, maybe your answer is, okay, it's the pathway respond to stimulant, right? But I want to have this, you need to screenshot your finding from Galaxy. So when you do a screenshot, please do not crop, right? You do not crop. I want to see entire screen of a screenshot, so that is a proof of your answer. If I only see you, if you only answer this, right, respond to temperature stimulant, but you did not show me the output of your Galaxy, right, then you will get no mark. So your answer is not, there's no standard answer, because every single of you having a different data set. Yes, you have to extract the data set from, you use the same tool to run. For example, if I ask you to assemble a genome, you use the same tool to run. I will mention that, okay, you need to use the ABIS to assemble the genome and blah, blah. And you need to use the DEC to run the differential express, right? But then you will have a different answer. So each of you will have different answer. And the mark only giving if you show the proof. If you only answer without giving the proof, that indicate that you don't understand the analysis itself, and you wouldn't get any single mark, even you answer it. So the proof you must attach together with your exam paper. Right, I will give a exam paper, then you can, in Word file, you download it, answer there, and attach your result, your output as a proof. And you have to clearly seeing that, put the screenshot correctly. Don't put let's say your screenshot for answer number 2 in answer number 1. Then I will not give mark. 1 hour. You have to complete it within 1 hour.
Doctor, since the test is online, so we don't have to be physically present, right?
Our test is 2 to 5, right?
Yeah, correct.
Yeah, 2 to 5. And last time we all physically present. I want you guys to be there.
So we will be at BT17, is it?
BT17, no, right? This practical, you guys, for what GBM or BGM or something? Is it there?
Yes, at the
The lab.
Yeah, I think we will be there. Yeah.
Okay.
So 2 o'clock. It's online,
Okay.
because you have to use online tools. You have to use Galaxy to do your test. But I want you guys to physically present there. Is it okay?
Can, doctor.
All right. If, for example, if we have an issue, right? Let's say during the day, the line is not good, right? Then I would need to do the test again. Maybe that time I will use, you can do it at home, right? But then I will have to change change the question. But you see, we have at least 2 hours, from 2 o'clock to 4 o'clock, right? We must finish. So the analysis won't be many. There are only I think 3 or 4 questions. Yeah. And each run, it should be quite fast one. Yeah. I maybe ask you guys to upload the data set first, right? To to Galaxy. then I will give instruction how you extract the data from. Now all the students will have one original data set, right? And then each of you will have to extract your own data set from the original data set, right? Then you get a sub data set and you analyze based on the sub data set. Okay. Yeah. So regarding the line, I think Sophia, you mentioned the line in there is already bad, or the line in the GBM1 or GBM2, is it very bad?
It's actually quite bad. Very, very bad.
So you cannot connect to any internet or something?
We usually cannot access the Wi-Fi.
So it's the Wi-Fi issue. How-
Line also lah. You call also, you cannot receive one.
Okay. How about BT 17 or BT 18?
When there's a lot of us, the line becomes very bad.
In BT 17 or 18?
Yeah, when a few people are there, it's okay. But then when all of us are there, then the line becomes very bad.
Okay. Okay, then let me think how to mitigate this issue. Where can we do? But I think any place in UN is still the same. No matter where you go, all the internet is very bad. Ihan, if you have time, right, can you go to the office today to check whether any BT 17 or BT 18 is free?
Next
Tuesday, 2 to 4.
Okay, doctor.
Yeah. Check if possible, if both room is empty, right? Then it will be good. We can split the group into one, some is 18 and some is 17.
Okay, doctor.
Yeah. Let me know. Yeah. If cannot, then then we have only to do it online. Yeah. But then if you want to do it online, I think the question will be more tougher. Right. Okay. All right then. Any more questions for the practical? Please. Yeah. Yes.
For the practical, so for the practical report,
Yes.
currently the due date is next Tuesday. Can we ask for extension?
Next Tuesday, yeah. Next Tuesday is okay. Then I'll change to next Friday. Can I? Because I need the time to mark. It's quite many of you.
If next Sunday can.
Sunday, because the week after I have to submit already.
Oh, okay.
Yeah, because
Friday.
because this is continual assessment, right? You don't have a test for this practical, right? So I need to submit the report to them, to the course coordinator. I think it's Dr. Ariana or something, earlier as possible. And I haven't marked also others. Your 2019, your midterm test, and also the assignment. So I will need time to mark. Yeah, that's why. You don't need to write very long. I mean, as long as your explanations, your result explained properly, your discussion point have citation, and I think it should be okay. Yeah. Yeah, basically it's only 10 pages. Not more than 10 pages, sorry. Yeah. Including the reference. All right. Okay. Yeah, we'll try. I mean, we'll start 1 hour first. If you can finish in 1 hour, we'll do it in 1 hour. If you cannot finish in 1 hour, then we'll do it in 2 hours. Right. But 2 hours is the max. Because if not, then it will become like final exam, final exam pun 2 hours only. You want to do it through 3 hours. I don't want too long. Right. Okay. Any more questions? No. Okay. Then I think that's all for today. All right then. Thank you guys. That's all for today.
Okay,
Thank you,
Doctor.
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 All right. Please practice yourself. Practice
Thank you, Doctor.
All right. Okay. Thank you. Thank you.